Hello, Indiana Virtual students. As promised, here is a video to help you through the assignment 0305 DNA replication. Okay, so as always, we have the link here to the assignment. Okay, and so there is your template of what you will use for this assignment, and we have our grading rubric. Okay, so for this lesson, I'm actually going to go over to our website, and I am going to our Google site website, that is, and I'm going to scroll down to lesson 0305 over here, because as always, I have created the Google slide for you of the template, but I've added a little bit of extra help on the template over here, so I'm really going to recommend that you use the template that I've created for you. Okay, so you have already gone through the lesson, you've taken notes, you've learned all about DNA replication, transcription, and translation. Okay, so as a little reminder here, little synopsis of this lesson, said in this lesson you learned about the structure of DNA and how DNA is copied. Hopefully now you are able to compare the relationship between DNA and RNA, and hopefully now you feel better equipped to walk through the processes of transcription and translation. Okay, so we will need to follow those steps of DNA replication, transcription, and translation to ultimately make a protein molecule. So use the following DNA replication assignment as a template to complete this assignment. Okay, additionally, be sure to check out the grading rubric to make sure you have everything done that's needed. Okay, so I'm going to open up the Google Doc here, okay, and then we have some instructions. Okay, for this assignment, you will replicate DNA to make a complementary strand of DNA. Then you're going to transcribe the complementary DNA sequence into mRNA. Next, you will translate the mRNA molecule into the codes for specific amino acids using the amino acid chart on the last page of this document. And lastly, address the reflection questions. Okay, so your whole lesson that you learned about through all of 3.5 was about how DNA is copied in the process of DNA replication. So we copy DNA to DNA. And then how DNA can go through the process of transcription, where DNA is copied into a strand of mRNA, and then that mRNA is translated into an amino acid chain. Okay, the whole purpose of DNA replication is to make copies of DNA. So say we have mitosis or meiosis going on, we need extra copies of DNA. We're making new cells, so we need more DNA. And you hopefully remember that in the interphase stage back in some of our previous lessons. Okay, now transcription and translation, the purpose of transcription and translation is protein synthesis. You know, our cells are constantly making proteins. So DNA is the ultimate instructions to build those proteins. And so we take DNA, we copy it into a single strand of mRNA, that's transcription, and then we use that mRNA out at a ribosome to actually translate it into an amino acid chain, and an amino acid chain is a protein. So I'm going to do some of this lesson with you, and then hopefully with what I share with you here, you'll be able to continue on your own. Okay, so first thing we need to do is copy DNA. We need to replicate DNA. And if you need some extra help with this lesson, you can go back to page 3, slide 3 of lesson 3.5. Okay, so here's our original DNA. Okay, and what we need to do is make a complementary strand. Okay, so something you have to remember back in the lesson is A always pairs up with a certain base, and T always pairs up with a certain base, G always pairs up with another base, and C always pairs up with a base. So one of the rules you have to remember is the complementary base pair rule. Adenine always pairs with thymine, and guanine always pairs with cytosine. So the complementary DNA here is A pairs with a T, T pairs with an A, and G pairs with a C. So what you'll do is continue on and filling in these bases as you go. So whenever you see a C, you're going to put a G. Whenever you see a T, you're going to put an A. Okay, because A always pairs with T, C always pairs with G. Okay, now it says to transcribe the DNA to make an mRNA molecule. And again, if you need help on transcription, go back to slide 5 of the lesson. Okay, so one thing that we have to do though is we have to copy our complementary strand of DNA down here. So we're literally going to copy that strand down here. Okay, and I'm just going to do those first 3 bases with you. Okay, now when we go through transcription, we have to remember that we are copying DNA into a strand of mRNA. So we have to be aware with what matches up with what. So DNA contains the bases A, T, C, and G, while RNA only contains the bases A, U, G, and C. So we have to be careful with what pairs up with what. Okay, so whenever we have a T in DNA, it's going to pair up with an A in mRNA. Okay, whenever we have an A in DNA, here's where we have to be careful. Normally an A would pair up with a T. However, there are not T's in mRNA. So this is where U, uracil, comes in instead. And then C pairs up with a G. All right. Later on, you may have a G that's in your complementary DNA, and a G is going to pair up with a C. Okay, so let me get rid of those because those two don't actually go there. All right, so now we have our mRNA molecule. Okay, so now something else to think about, this DNA replication, that took place in the nucleus. Okay, transcription took place in the nucleus. Okay, but now this mRNA molecule is actually now going to leave the nucleus and go out to the cytoplasm, to a ribosome, to be translated into an amino acid chain. So now what we have to do is we have to copy our AUG and add that down here. Okay, and now we're going to translate the mRNA into a corresponding amino acid sequence here. Okay, if you need help with translation, you can go back to slide 6 of lesson 3.5. Okay, now to do this, we actually have to scroll down here, and we need to use this chart. Okay, and actually, remember, we've got to remember this, AUG. We're going to look up AUG. Now, this chart is, and I'm just going to jot this here for a second, AUG, so we remember. Okay, this chart is an mRNA codon chart. Okay, what we have here, this AUG, is a codon. A codon corresponds to an amino acid on this chart. So when we're using this square chart, and there's another chart I'm going to show you in a second, in using this square chart, we're going to find the first letter of the codon here. Okay, so our first letter is an A. Okay, so here's the A. So far, we're going to work within this large row right here. Okay, what we're doing is we're looking for AUG on this chart. So could you just go and look around until you find AUG? You could, okay, or you can use this method and how this chart works. Okay, so we're going to be in this row for the A. Okay, now we go to the top. This is the second letter of the codon. Okay, our second letter is a U. So now that's narrowed us down to, excuse me, that's narrowed us down to this row, or this column and this row. So we can only look within this square because that's where the column and the row overlap. Okay, so now over here we go to the third letter of the codon. So now our third letter is a G. So now our G's right here. We are only in this final section here. So we have to look to see where does this row, this column, and then this little row all overlap. And there it is, stands out in orange. AUG stands for methionine, which is our start codon. Okay, and we can abbreviate these. We can abbreviate them by their first 3 letters. Okay, so methionine. Okay, now I'm also going to show you this chart here. A lot of students really like the circle chart instead of the square chart. Okay, what you do is you start in the center, you find your first letter. So here's our A. Okay, and then you go to your second letter. Here's our U. And then you go to your third letter, there's our G. And so it stands for MET, M-E-T, which is methionine. Okay? All right, so let me delete that. And what we're going to do is go up here, and now we're going to document that our amino acid is methionine. Okay, so now what you're going to do is go back to the beginning, and you're going to continue your DNA replication. Once you have your entire DNA replicated, make sure you bring it down here as well. Then go through transcription and complete the next row. Okay, copy that mRNA molecule down here to the third section, translation, and then you're going to use the chart to look up the following amino acids here. Okay, once you have all that done, then you're going to do it all again here. Okay, you're given a new strand of DNA, copy it into the complementary DNA, then with that complementary DNA, find the corresponding mRNA molecule, and then use the chart again to look up the amino acids. Okay, and lastly, you have some post-lab questions or post-activity questions. Okay, and there's some hints there where to find some answers for this. Alrighty, guys, so please send me emails, send me questions if you struggle or if you have any concerns or you don't know what to do. Reach out to me. All right, thanks, guys. Bye.